Today I will be discussing about bacterial growth kinetics. Bacterial growth kinetics is the study of how bacteria proliferate and multiply over time under various conditions. This field is crucial for understanding the growth patterns of bacterial populations, predicting their behavior and developing strategies to control their growth. Bacterial growth kinetics involve the quantitative analysis of bacterial growth rates, population size and factors influencing growth. Bacteria are single celled microorganisms that can replicate rapidly under favorable conditions. The study of their growth kinetics help us comprehend the dynamics of their growth in environments such as laboratory cultures, industrial processes and natural ecosystems. Bacterial growth is influenced by a range of factors that collectively determine how quickly and successfully bacteria can multiply and thrive. These factors play a crucial role in shaping the dynamics of bacterial populations and their interactions with their environment. Let us look into some key factors that affect bacterial growth. Here you can see the temperature, pressure, light, oxygen, pH and carbon. So, there are so many factors are there for bacterial growth. First one is nutrient availability. Adequate nutrients including carbon, nitrogen, phosphorus and various trace elements are essential for bacterial growth. Bacteria require these nutrients to synthesize protein, DNA and other cellular components. A lack of essential nutrient can limit growth. Second one is the temperature. Bacteria have specific temperature ranges at which they thrive. The optimal growth temperature varies among different bacterial species. Too low or too high temperature can either slow down growth or denature cellular components, leading to cell death. Third one is pH level. Bacteria also have specific pH ranges that support their growth. Changes in pH can impact the ionization of cellular components and enzymatic reactions. Bacteria adapted to acidic environments like certain food spoilage bacteria differ from those thriving in a alkaline conditions. Next factor is the oxygen availability. Oxygen plays a critical role in bacterial metabolism. Bacteria can be categorized as aerobic, means the bacteria requiring oxygen, anaerobic means thriving in the absence of oxygen, or facultative anaerobes, that means they are surviving in both the conditions. Oxygen availability affects energy production and influences growth patterns. Next factor is the water availability. Bacteria need water for various cellular processes. Environmental conditions with low water availability can hinder bacterial growth by limiting nutrient transport and cellular activities. Next factor is osmotic pressure. The concentration of solutes in the surrounding environment affects bacterial growth. High osmotic pressure caused by high salt or sugar concentrations can lead to water loss from bacterial cells and hinder their growth. Inhibitory substances also one of the factor. Some environments contain substances that inhibit bacterial growth, such as antibiotic or toxic chemicals. These substances can directly affect bacterial viability and reproduction. Next is competition and cooperation. Bacterial growth can be influenced by interactions with other microorganisms in their environment. Competition for resources or the release of growth promoting compounds by neighboring bacteria can affect the overall growth rate. Genetic factor, most important factor. Bacteria with genetic mutations or adaptations can exhibit different growth kinetics. Some mutations may confer advantages in specific environments, leading to variations in growth rates. Quorum sensing. Many bacteria communicate with each other through quorum sensing. It is a process that regulates gene expression in response to cell density. This communication can influence growth patterns and behaviors. Understanding these factors and their interactions is essential for controlling bacterial growth in various contexts, such as food production, healthcare and environmental management. By manipulating these factors, students can develop strategies to either promote or inhibit bacterial growth as needed. The growth kinetics of bacteria refer to the patterns and dynamics that govern the proliferation of bacterial populations over time. This fundamental concept is central to microbiology, offering insight into how bacteria respond to changing conditions and interact with their environment. Bacterial growth kinetics can be broken down into distinct phases, each contributing to the overall understanding of bacterial population dynamics. You can see in this pictorial representation lag phase first, then log phase, then stationary phase and death phase. In lag phase, at the beginning of the growth process, bacteria undergo a lag phase. During this phase, they are adjusting to their new environment, preparing their metabolic machinery and not at actively dividing. The duration of this phase varies depending on factors such as the bacterial species and conditions of their surroundings. Second phase is the exponential or log phase. Once bacteria have adapted to their environment, they enter in the exponential phase. In this phase, bacterial cells divide rapidly and exponentially, resulting in a substantial increase in population size. This phase is characterized by a consistent and maximum growth rate under optimal condition. Third phase is the stationary phase. As time goes on, the availability of nutrients and space in the environment starts to decline due to bacterial growth and metabolism. This leads to a decrease in the growth rate and the population level off. This phase is known as the stationary phase, During this phase, the rate of cell division balances out with the rate of cell death, resulting in a relatively stable population. Fourth one is the death or decline phase. Eventually, the environment becomes less favorable for bacterial growth. Nutrient depletion, accumulation of waste product and other factors contribute to a decline in a growth rate. As a result, the bacterial population enters the death phase. During this phase, the number of viable bacterial cells decreases due to cell death and new cell division. The growth kinetics of bacteria are influenced by various factors as discussed previously, including the availability of nutrients, temperature, pH, oxygen level and other environmental conditions. Understanding these factors and their impact on growth kinetics is essential for fields like microbiology, medicine and biotechnology. Mathematical models have been developed to describe bacterial growth kinetics. These models help students predict how bacterial population will change under different conditions, aiding in the development of strategies for managing bacterial growth, where the industrial setting, healthcare or environmental monitoring. The Monod equation is a mathematical model that describes the growth rate and microorganism in a culture as the function of the concentration of the limiting nutrients. It is commonly used in microbiology, biotechnology and environmental science to predict the growth of microorganism like bacteria and yeast in a controlled environment. The equation is named after its developer Jacques Monod, a French biologist who made significant contribution to our understanding of microbial growth and enzyme regulation. The Monod equation is typically written as follows: mu is equal to mu max into S upon Ks plus S, where mu is the specific growth rate of the microorganism. Mu max is the maximum specific growth rate of the microorganism under optimal nutrient condition. S is the concentration of the limiting substrate or nutrient in the environment. Ks is the Monod constant, where it represents the substrate concentration at which the specific growth rate is half of the maximum rate. The Monod equation suggests that the specific growth rate of microorganism is dependent on the availability of a specific nutrient in their environment. As the substrate concentration S increases, the specific growth rate mu approaches its maximum value, that is mu max. The Monod constant Ks reflects the affinity of the microorganism for the substrate. A smaller indicates a higher affinity for the substrate. The equation implies that when the substrate concentration is much higher than the Ks, the growth rate becomes nearly constant and equal to mu max. However, when the substrate concentration much lower than Ks, the growth rate become proportional to the substrate concentration. The Monod equation is widely used in various fields, including bioreactor design, wastewater treatment and biotechnology processes. It provides insight into how microorganisms respond to changes in nutrient availability and helps in optimizing conditions for desired microbial growth and product formation. Other mathematical models such as the logistic growth model and the Gompertz model has also been developed to describe and predict bacterial growth kinetics. These models help students, researchers and understand how different factors impact bacterial growth and design strategies to control bacterial population for various applications. The logistic growth model, it is a mathematical framework used to describe the population dynamics of organism including bacteria as they grow and interact within a limited environment. This model provides a more realistic depiction of the growth than exponential model by incorporating factors such as resource limitation and carrying capacity. The logistic growth equation is typically expressed as dn upon dt is equal to r into n into 1 minus n upon k, where dn upon dt responds or represents the rate of change of population size, that is n, over time t. R is the intrinsic growth rate which represents the maximum rate of population increases under optimal conditions. K is the carrying capacity of the environment which denotes the maximum population size that the environment can sustain. At low population size n, the growth rate is close to the maximum rate, that is R, resembling exponential growth. As the population size approaches the carrying capacity k, the term 1 minus n upon k approaches 0, causing the growth rate to decrease. When the population size equals the carrying capacity, the growth rate becomes 0, indicating that the environment's resources are fully utilized and growth levels off. In the initial stages, bacterial population experience rapid growth due to abundant resources. As the population increases and resources become scarcer, the growth rate slows down. Ultimately, the population reaches a point where the growth rate matches the death rate, resulting in a stable population size near the carrying capacity. The logistic growth model is valuable for understanding and predicting population dynamics in various fields such as ecology, epidemiology and microbiology. In bacterial culture, this model helps anticipate how growth rate will change as resources become limited, guiding decisions in bioprocess optimization and microbiological research. The logistic growth model provides a more realistic representation of population growth by considering the interplay between resource availability and carrying capacity. It highlights how bacterial population respond to changing environments and provides insight that contribute the various scientific and practical applications. The Gompertz model. The Gompertz model is mathematical framework used to describe the growth dynamics of bacterial population in a more redefined manner than simple exponential or logistic models. Developed by Benjamin Gompertz, this model considers the gradual slowing down of growth as resources become limited and population approaches a satisfactory stationary phase. The Gomper's equation is typically expressed as n t is equal to n 0 into exponential minus exponential mu max into E upon n 0 into lambda t plus 1, where n or t represents the population size at time t, n 0 is the initial population size, mu max is the maximum specific growth rate, and E is the base of natural logarithm approximately 2.71828. Lambda is the lag time indicating the time it takes for the population to start growing significantly. The Gomper's model is particularly useful when bacterial growth exhibits a lag phase before exponential growth and gradual decretion. It accounts for the asymmetry in growth curves where growth rates during the initial and later phases differ. The Gomper's model finds application in fields such as microbiology, biotechnology and medicine, where accurate predictions of bacterial growth patterns are essential. It is valuable for understanding complex growth dynamics in bacterial culture, such as those involving adaptation periods or changing environmental conditions. In summary, the Gomper's model offers a more detailed representation of bacterial growth dynamics by considering the lag phase, exponential growth and gradual decretion. This model is beneficial for studying microbial behavior in situations where growth patterns are more intricate than those described by simpler model. The Gomper's model captures both exponential growth at the initial phase and a gradual decretion as the population size increases. The exponential term inside the outer exponential function approaches 0 as time t, approaches the lag time lambda, signifying the outset of significant growth. As time progresses beyond the lag phase, the exponential term inside the outer exponential function becomes larger, causing the growth rate to slow down. Different methods of measuring bacterial growth. Measuring bacterial growth is a fundamental aspect of microbiology that helps students understand microbial behavior, study their interaction and optimize various applications. Various methods are employed to quantitatively assess bacterial growth. Let us now look into the several common approaches. First one is plate count method. This plate count method involves diluting a bacterial sample and spreading it into an agar plate. After incubation, individual colonies representing viable bacteria cells are counted. This method provides an estimate of the colony forming units CFUs per unit volume of the original sample, that you can see in the pictorial representation. Here in the pictorial representation also you can see the turbidity measurement, that is optical density. Measuring the decrease in the light transmission through a bacterial culture indirectly reflects its turbidity, which corresponds to cell density. Optical density or OD is typically measured using a spectrophotometer at a specific wavelength. A higher OD indicates higher cell density and growth. Third one is the viable cell count. Viable cell counting involves directly counting individual viable cell using microscopy, automated cell counters, streamlined this process by using image analysis algorithm to quickly count cells in a sample. Fourth one is dry weight measurement. For certain bacterial culture, the biomass can be estimated by measuring the weight of dried cells after removing moisture through drying. Fifth one is the direct microscopic count. Using a specialized counting chamber and a microscope, the student can directly count the number of the cell present in a known volume of sample. This method provides rapid result, but may not differentiate between viable and non-viable cells. Then ATP measurement. ATP is a molecule found in all living cells and serve as an indicator of cellular activity. ATP measurement system detect the amount of ATP in a sample, which can then be correlated with bacterial cell count and growth. Seventh one is the metabolic activity measurement. Certain methods assess bacterial metabolic activity to infer their growth. Next one is the nucleic acid quantification. Quantifying the amount of bacterial nucleic acid, DNA or RNA in a sample can provide the estimate of bacterial biomass and growth. Then bioluminescence assay. Bioluminescence bacteria emit light as a result of metabolic activity. Next one is the microbial impedance measurement. This method involves measuring the impedance of resistance of culture medium. The choice of method depends on the specific goals of the study, the type of bacteria being studied and the available resources. Combining multiple growth of kinetics is a fundamental concept that underpins our understanding of how bacteria proliferate and interact with their environment. By studying the bacterial growth kinetics encompasses the study of how bacteria proliferate and interact with their environment over time. Thus, this concept is crucial for understanding microbial behavior, optimizing processes and making informed decisions across various scientific and practical domains. Here is a concise summary. The phases of growth, as we know, the lag phase, log phase, stationary phase and death phase. And then factors influencing growth: nutrient availability, temperature, pH, oxygen level, water availability, osmotic pressure and inhibitory substances. Then different measurement methods, various methods quantify bacterial growth. Then mathematical models, different models I have explained. Then applications: bacterial growth kinetics have application in diverse fields like medicine, microbiology, biotechnology and environmental science. They aid in disease treatment, bioprocess optimization, food safety and waste management. Understanding bacterial growth kinetics provides insight into how bacteria thrive, adapt and interact with their surroundings. This knowledge empowers students and professionals to harness microbial processes for beneficial outcomes and make informed decisions in a wide range of contexts. Thank you.